Hi, my name is Sienna Vogel, and today I'll be presenting on gene therapy. More specifically, we'll look at gene therapy as a possible future treatment for cancer. So what is gene therapy? Gene therapy, in short, is the introduction of genetic material to a cell. This can either be because it's compensating for abnormal functioning, or because it needs help in making proteins. The type of proteins that it can aid in making are the ones that we studied when looking at DNA and RNA. Currently, gene therapy is being studied as a way to treat or cure diseases. This can be done in three ways. Scientists can replace a disease-causing gene with a healthy copy of the gene. They can inactivate a disease-causing gene that is not functioning properly. As we know, genes can be turned on and off. Or they can introduce a new or modified gene into the body to help treat a disease. As you can see in the bottom left-hand corner, I have a little diagram posted. This is showing the introduction of a new gene into the body. So as we can see, the cell is not functioning with the gene. It gets introduced into a new functional version of the gene, and then the cell can go on functioning normally. So how does gene therapy work? A carrier called a vector is used to transport a therapeutic gene directly into the cell. Currently, the most common vector is actually a virus. Scientists will genetically modify viruses to carry human DNA and then inject this virus into cells, whether inside the body or outside the body. Once the target cells are infected with the vector, it releases the new genetic material. This genetic material then creates a functional protein product that enables the cell to return to a normal state and thusly normal functioning. So there's two major types of gene therapy. We have somatic gene therapy and germline gene therapy. We're going to start by discussing somatic gene therapy because all current research on correcting gene deficits is in somatic cells. So somatic cells are any cells that are not used for reproduction. So somatic gene therapy is transferring therapeutic genes into these cells. So these can be bone marrow cells, blood cells, and skin cells, just to name a few. The functioning gene that was just injected, however, cannot be passed on to offspring. On the other hand, we have germline gene therapy. Now, due to ethical reasons, germline gene therapy is not currently being attempted and it is not widely studied. Germline cells are any cells that are used for reproduction. So these include egg and sperm cells. Therefore, germline gene therapy is when therapeutic genes are transferred into these germ cells. Germ cells are passed to offspring. Therefore, if you inject an egg or a sperm with therapeutic genes, it'll get passed to the child. There's also two types of gene delivery. We have ex vivo delivery and in vivo delivery. So to begin with ex vivo, I like to think of ex as in external. So this happens outside of the body. So cells are first taken from the patient. They are collected and placed in a petri dish and stored in a lab. Scientists will then add the new therapeutic gene material, and once the cells begin to function normally again, they'll be reinserted back into the patient with the new functioning cells, and sometimes they're reinserted by the millions. These are frequently used to treat blood and immune diseases because the circulatory system and the immune system, it's very hard to target one specific cell. So by inserting millions or hundreds of them, they'll overtake the cells that are not functioning correctly. We also have in vivo delivery. This is a direct transfer of gene into a target organ or tissue. So this can be sometimes dangerous because the target spot can be missed. So now let's talk about the scholarly article that I researched, which was all about treating cancer with gene therapy. So T cells are vital to our immune system. As you've probably studied in a high school biology class, T cells identify the healthy cells from the unhealthy cells, in this case cancerous cells, and then they attack. However, because of thymic selection, only about 2% of T cells produced survive. which leads to a low count of T cells in the human body. So, my article posed the question that could genetically altering T cells by introducing CARs and TCRs result in the killing of more cancer cells and the stoppage of tumors? So, over on the left-hand side again, I have a diagram that just explains this process. So, the T cells are acquired through a patient's blood. You then insert the gene, whether it's CAR, TAR. Then you create a bunch of them, grow them in the petri dishes, and then insert them back into the patient. Also, I have the citation of the scholarly article in the lower right-hand corner. So T cell modification. So my article said that when the CARs were added, the T cells started to act like antibodies and they started to target surface tumor antigens. When TCRs were added, they were able to better identify neoantigens, which is basically any mutated antigen. In order to do this, they sequence a patient's cancer cells, identify cancer-specific mutations, and then they target them. So due to these modifications, I found this beyond interesting. Anti-tumor responses were seen in 50% of patients, and 80% of patients showed clinical benefits. So I bet you're thinking to yourself right now, why isn't everybody using gene therapy as a treatment? It works so well in a disease that has killed many throughout the years. However, there's lots of associated risks with gene therapy. Also, on the right-hand side, I have a little chart that just talks about the CAR T cell cancer therapy. And basically, it's the same thing as ex vivo. So you take out the genetic material from blood, alter it, and then infuse it back into the patient. So, as I mentioned, gene therapy does have some downfalls. As of right now, the long-term safety of gene therapy has not been researched. It's a fairly new practice, and a lot of people do have ethical issues with it, so it's not really practiced widely. The current gene therapy methods may target the wrong cell. As I talked about in prior slides, if you could get injected in the wrong spot, which could lead to a tumor. Also, there could be unwanted immune reactions because your white blood cells could see the virus for what it is, a virus, target, attack it, and that way the genetic material is no longer. So not only are you down a treatment, but you're also down millions of dollars, which it also costs to have gene therapy. So in conclusion, gene therapy presents a new experimental approach to treating and stopping disease. Gene therapy is commonly performed by injecting a modified virus into a targeted cell. Genes can be modified both outside of this body, ex vivo, and inside of the body, in vivo. While it could offer a possible treatment for diseases such as cancer, research is not comprehensive and there are many associated risks. So to leave you, I have a few questions to think about. So number one, what is the most commonly used vector? And remember, a vector is what transports the therapeutic gene material into the cell. Number two is what's the difference between ex vivo delivery and in vivo delivery? And finally, as I talked about with our scholarly article, what type of cells were genetically modified to treat cancer? So I'll let you guys think about that. In conclusion, I have some of my sources. Thanks for listening and good luck with those questions.